Welcome everybody. Thanks to attend today's episode of tinyML Talks. We have the pleasure to have as a speaker Martin Peacock, Director and Chief Scientific Officer of Zimmer and Peacock. The title of his speech is Biosensing at the Edge of the Cloud. It will be a great pleasure to have Martin time to be with us, and let's hear from him. But before to leave the floor to Martin, let me remind this type of event can be organized thanks to tinyML Foundation, obviously, and thanks to all the strategic partners which make this event and many other events under the auspices of tinyML Foundation true. And let me remind what are the strategic partners. So they are AI Zip, Analog Devices, Arduino, ARM, BrainChip, Edge Impulse, Efficient, Green Waste Technology, Gravity, IMAX, IBM, ImageMob, Infineon, Innatera, Microsoft, Nota AI, NXP, Polytechnology, Quixo, Qualcomm, Renesas, Schneider Electric, Sensimil, Sony, Silicon Labs, ST Microelectronics, Synaptics, Syntiant, TDK. So as you can see, our strategic partner list is growing from time to time. This is a great sign of the community being active and receiving more and more support. There is a great amount of content and opportunities within the tinyML community that are sitting ready for us. Two types of things I would like to remind. The meetup are a great events that can be organized at local level and between the members of the community, such that there are opportunity to share and to put in common the knowledge and the activities such that more opportunity can arise. And it is outstanding how many groups, we are close to 50 in 41 countries, how the meetup are really taking place with success. The other mean of the content of the community is the LinkedIn page, through which the tinyML community also exchange information, news, contributions, which is growing without pace. And today we are 4K members with close to 13K followers. In terms of content, the YouTube channel of tinyML Foundation is a great place where to learn. Essentially, there are more than 600 videos, lot of content from all the activities that are taking in place in tinyML Foundation, and therefore you will have your opportunity to take the content that interests you and to learn from it. There is no equivalent all around the world, so please take advantage of it. Then let me remind that tinyML Asia Technical Forum is going to take place on November 16 this year in Seoul, South Korea. This is a in-person event, so please join as many as you can to this event, which is focused on the Asia region. And the content, in the case you will miss in-person event, will be shortly available after the conference, let's say in 1 week or so that you can enjoy all the video available. A great content that has been made available for the very first time by the tinyML community, the tinyML Foundation and community, has been the 2023 Edge AI Technology Report. Just to remind you the success of this report, in just 2 months, there were 10K downloads. So all the companies, many companies contributed to the content. The content is really spanning from software, hardware, different technologies, and these are helpful, obviously, to the tinyML community, but also to the young professional, to really interface to this new exciting world, which is the tinyML world, for the very first time. The report will be also upgraded on 2024, and during Q1, let's say at the end of Q1, initial Q2, this will be there will be a new version available. So stay tuned for these progresses talk. So short bio about Martin. Dr. Martin Peacock is an industrial bioelectrochemist with over 20 years of biosensor experience, having had industrial roles from Abbott Diabetes to GSK, and solving technical challenges from continuous glucose monitoring to RNA analysis. In recent years, Martin has set up a biosensor-focused company across the globe, from Silicon Valley, California, to Oslo, Norway. So I'm releasing the control. Martin, thanks a lot for your availability, and looking forward to your great talk. Thank you.
Thank you, Danilo. I appreciate it. So thank you to the tinyML Foundation for inviting me to speak this afternoon. I just put this abstract up to essentially remind myself what we are speaking about. But as stated, rather than me just sort of monologuing at everyone, feel free to answer questions or ask questions in real time. I think I much prefer an interaction rather than the drone of my voice for the sort of next 45 minutes, let's say. But let me go forward. And I just want to start with a kind of call to action. What I mean by this is a little bit provocative, but I say here that all the easy stuff is done, and now it's time for biology. I've obviously paraphrased it and crunched it down a little bit, but I will explain this, as I go forward today, that, when I sort of tinyML and the Internet of Things, I feel like, we've done a lot of the sort of physics problems and a lot of the condition sensing, but I feel like the real value now going forward is in the biology, and I will expand upon that at the moment. And that really reflects the title of my talk, which is, sort of sensing at the sort of bio edge. So today I will be very quick. I'll introduce myself just very quickly, but that has been done, so I will be super quick. I'll explain why ZP exists, and really we kind of originally existed because of a phenomenon called the Internet of Things, and we kind of saw that there was a gap in the technology and the application space for that. So I will expand upon that. So we literally sort of day one, when we put up the sort of the landing page, IoT was in the Internet of Things, which was a movement that's been around for over a decade now, but we did see that this was definitely a sort of thing that was going to happen, and we saw a gap in the market and a gap in the technology, and I will address that. I will then get into the science and technology of what we do, and I'm going to talk about two sides of things. I'm going to talk about wearables. I'm wearing a wearable at the moment. This is configured to be a CGM, a continuous glucose monitor, and I will also talk about embedded sensors, but I also want to touch upon IVD, in vitro diagnostics. Once upon a time, you'd have to kind of explain to people what rapid diagnostic testing was, but unfortunately, COVID-19 kind of taught people the value of RTD, this rapid diagnostic testing, and I put a few acronyms up here: point of care diagnostics, spot testing, lab on a chip, IVD. These are all synonyms for, in my perspective, really for the same thing, which is take a sample, get a result, immediately give the result to that end user. And by the way, you can also store it on the cloud. And I'll talk about applications. We're working in food quality, we're working in clean water, and we're definitely working in health. So for me, the sort of tinyML community, I'm very, very new to your community, but I appreciate being invited along today. And I wonder how much tinyML is actually being applied to some of the things that we're interested in, which is, continuous glucose monitoring, nitrate monitoring, food quality. But I will touch upon these things as I speak today. So quick resume. I wouldn't say, my background is basically chemistry. So I've kind of migrated from chemistry to electrochemistry to electrochemical sensors into biosensors. And I actually thought nowadays, I do say to everyone, we all have to be data scientists. So I definitely feel that I am trying to become a data scientist, or at least at a high level, understand data science, at least, as a toolkit within the business. So I have a big, one of the comments I'll make today is actually these days we can make sensors that in the past we would have had to put all the effort into the sensors. Now I would say we'll put 80% of effort into the sensors, make them 80% good, but actually we'll close the gap with good data science. And we can discuss that today if the questions pop up. Very brief introduction to ZP. We were launched in 2014. Myself and my business partner, we are ISO 13485. So we understand how to navigate this medical wearables and this IVD space. And when I say medical wearables, we're really doing electrochemical sensing, and I will expand on that. We do contract development, contract manufacturing, and we're in Norway and UK. So hopefully that's quick enough. We have a ZP team. We're not huge. We're not small. 23 languages spoken. Like any modern company, you have to have, not have to have, we want to have engineers and scientists from all over the world. And we do have state-of-the-art facilities, and we have several facilities, as you can see, in Europe. So why are we here? So why I am here really is because I started my career in glucose strips. Now, this was obviously, well, not obviously, but glucose strips, have taught the world a lot of things. It's taught us that you can have distributed testing. You can have testing actually in people's homes. And these glucose strip technologies really started sort of even pre-internet. I think this kind of technology was around in the 80s, and we became electrochemical at the end of the 80s and at the beginning of the 90s, but it was a sort of pre-internet of things and pre-internet technology. But glucose sensing has taught us a lot, and it's taught us that you can take a drop of blood, get 5 seconds later, you can know the glucose level in there. Now, that's kind of where my kind of career started with. And a lot of what ZP does is say, okay, take that fundamental hypothesis that glucose strips and glucose technology is a robust, scalable science. And then what else can we do with it? Now, what else has happened, obviously, in at least that diabetes space? And, diabetes is part of this smart bio edge. This is the Abbott Freestyle Libre 2. So it's not our product, but it is my arm. So, this is really a translation of the IVD technology, that blood glucose measurement that's happening in the left screen. They've essentially translated the science onto the right-hand part of the screen. And that, in fact, I've got one here, these kind of CGM discs, which you see on people. They have a little filament that goes through the skin and is measuring the glucose. I will talk about that kind of technology, but for me, tinyML is part of a technology stack. And at the very, how can I say, beginning of the technology stack is actually a sample, be it blood or be it interstitial fluid. And then there's a whole series of technologies that get that into information. And tinyML sits in that technology stack. So why was ZP? And I did mention this earlier on, that ZP, when we first started, we thought, right, we're going to make biosensors and we're going to really be part of the Internet of Things. And as I was preparing this presentation, I did see a sort of smart device in the public swimming pool yesterday, which was really a flow sensor for measuring the flow into urinals. So for me, the tinyML community, you've kind of, you have solved problems around flow. You have solved problems around the measurement of mass. I'm sure leak detection has been worked upon. The measurement of current, how many current sensors there are in an electric car. Acceleration has been worked upon. Machine vision and the interpretation of that has been worked upon. Proximity, velocity or measurement of speed, temperature and humidity, these two, I don't know how many kind of IoT and tinyML applications are around temperature humidity, but everyone sort of there's lots of people working on that. Also acoustics, but the elephant in the corner on all of this is where's the sort of biological, chemical, biochemical sensors? And the honest answer is there's not available or readily available these types of sensors. And that's really the role of ZP, that we think that the really useful data and information out there is actually in the biology, biochemical, and chemical space. But in order to access it, you need a sensor. And, we tend to use electrochemical sensors. And the reason we use electrochemical sensors is actually they've already proven themselves to be quite scalable in, for example, blood glucose measurement. Now, these sensors, we can, if we look at our environment in total, our environment is kind of made up of, solids, liquids, and gases. I do understand that the biggest phase in the universe is plasma, but I'm thinking strictly more about our globe. And when I sort of talk about the liquid phase, well, what are the things we can measure in the liquid phase that are at this bio edge? So if somebody came to us and said, I want to measure something in the sweat, that's perfect for the kind of technology that we like at Zimmer and Peacock. So, lactate in the sweat would be a classic. People are very interested in measuring things in saliva. So hormones in saliva is something that, we've been challenged with, and we're happy to overcome it. Question that's come to us, can you measure things in the tear film? You can see there's really a repetition here that, in particular at ZP, we're most experienced with measuring things in the liquid phase, and we don't mind if it's urine, if it's interstitial fluids, we love it. And if it's blood, we like it. Blood is kind of an interesting one because it's one of these it's very opaque, it's full of molecules, but yet electrochemistry can prove itself to actually be able to measure in that complex matrix. What makes what we do actually possible? We are just the derivative of, things that have really happened in the electronics community, and we're grateful for it, that devices have become low power, so we benefit from it. Things have miniaturized. Something that's very core to us at ZP is a scientific instrument called the potentiostat. Well, I looked at your list of sort of platinum members or platinum sponsors, and some of those companies have worked on miniaturizing the potentiostat. So now we can do electrochemistry that would have otherwise been in our laboratory. Actually, we can do it in the field next to the patient or next to the agricultural applications. But I'll expand upon that. Something that makes ZP and what we do possible is obviously connectivity, cloud computing. The fact is that at ZP, every time we build a new application, the data always goes to the clouds because it just opens up. It helps in so many ways for us. We will deliver the data there and then to the user, so they can look at their smart device, which will probably be in Bluetooth connection with one of our electronics, and they'll get a result. But at the same time, we will send the data immediately to the cloud because it really helps with quality control. It really helps with the business models, to be frank with you, to have data in the cloud, because then you open up that subscription model. We've really benefited from the explosion in data science over the last decade, I would say. But certainly over the last 5 years, it feels like there's been a real acceleration. So once upon a time, to make a electrochemical biosensor, you would have made the world's best biosensor. Now you can make a good biosensor, but good data science can help you actually plug that gap. And we've really benefited from smart devices. So tinyML, the Internet of Things, we're really all part of there's a trend, at least in the medical space, for miniaturization, that once upon a time, blood tests were all centralized. Great. Well, actually, people don't believe that centralization necessarily of testing is a good thing. So, there were tabletop devices. These became handheld type devices. These became almost palm-sized devices, and now they've become miniaturization. So there is a perceived benefit from taking tests that were otherwise done in the clinic and actually shrinking them down to such a state that actually now, it is a wearable device. And the CGM, the continuous glucose monitoring space, is the kind of poster child for wearable electrochemical biosensors. You've got some of you will be in San Diego, so you've got sort of Dexcom. You've got Dexcom down in San Diego. Some of you are in the kind of LA region, so you've got Medtronic in the LA region. And some of you are in the Valley, Silicon Valley, and there you've got Abbott there doing so. it's just to say that miniaturization of biosensors, and in particular electrochemical biosensors, is a thing, and it supports quite a large industry, which I'll touch upon. I'm just going to read this question very quickly. Yeah, so this is a good question. So the question that's just come through is, do sensors drift? And the quick answer is yes, they do. How do you fix it? Is the next question. Well, you fix it in part through good algorithms. So there is a place, and I think people were using, I want to say machine learning and tinyML, at least in some woolly way, not maybe calling it that, but actually they are using it in probably CGM today. In the first 24 hours, a CGM on somebody's body is probably drifting. Are they compensating for it? Yeah, sure they are. Are the algorithms deeply complicated? No, they can't be. So the quick answer is yes, electrochemical sensors drift. And so there is no perfect sensor out there. I do get this quite a bit, if I use piezo sensors, do I use photon-based sensors? Do I use electrochemical sensors? There is no perfect sensor. Academia is spinning its wheel trying to find the world's perfect sensor. And I'll probably touch upon that drift aspect actually in a minute as well. But we're focused on electrochemical sensors, and they have multitude of applications. We're using them in food quality, and I'll touch upon that. We're using them in agriculture for measuring the nitrate in the soil, and I'll touch upon that. We're using them in veterinary, a very challenging application, which tinyML can help with, because we're putting the entire sensor package in a fish, and the fish is alive, it's swimming around. So you can imagine all the kind of artifacts that are coming into our raw data, and you have to be able to separate out, for example, is this glucose or cortisol, or is this the fact that the fish is swimming? So there's some interesting challenges there. We can play them into horticulture, wearables, and we haven't forgotten that, electrochemistry and electrochemical sensing is really good for just finding analytes in quite dirty samples, including blood, which is a fairly complex sample. The reason people find us, and the reason that we've started this company, is because we thought, let's make a menu of electrochemical sensors. And this is, by the way, not our complete menu. But at Zimmer and Peacock, we started off by making electrochemical biosensors. So we make a glucose sensor. It's enzyme-based. It uses a technology called amperometry. We make an oxygen sensor. It's not enzyme-based. It uses amperometry. The oxygen sensor was, by the way, invented in 1956 by Leyland Clark. The value that we may be adding is its application into new space, and the fact that we've made it very small, very robust, whereas Leyland Clark in 1956, it looked like a pH sensor. We have a sensor for pH. That's a potentiometric measurement, chloride potentiometric. So what you find here is we have a whole list of analytes, and the electrochemical methods that may be running those sensors depends on things like, is it an ion, potassium, sodium, calcium, these are ions, pH. Then we tend to use potentiometry. Is it a small molecule? If it's a small molecule, we can sometimes detect them directly, or sometimes we use an enzyme. If it's an enzyme, we tend to be using amperometry. Sometimes if it's just direct, we may use voltammetry. I'm just going to read a couple of the questions. I must admit, one of them is about cyborg insects. Says one of the questions is, are electrochemical sensors affected by the drift, which I've answered. And then it says here, non-specific binding poses a significant challenge to accuracy, particularly with real blood samples. And I'm glad that that question's come up, because I'm going to show you in a minute a glucose sensor, and I'm going to show you that already there's mitigation in place to take care of things like non-specific binding. So I'll give you a real world example of that. But if I was to say about this slide says, if somebody was asking the question, can electrochemistry detect ions? The answer is yes. You could go to the ZP website and see that. Can electrochemistry do small molecules like ketones? Yes, go to the ZP website and you'll actually find, the raw data. Can it do macromolecules like troponin I, which is a marker for cardiovascular disease? Yes. We did do a patent and a technology for COVID-19 or SARS-CoV-2. It was a binding assay, so we can do pathogens and things like E. coli as well. And often we actually do it, if you can see my camera, we often do it in this form factor, which is a kind of, it looks like a glucose strip. But as I said, one of the theses of the company is actually to take scalable manufacturing techniques, which have proven to be usable with very non-ideal samples, and then but use it to test everything else but glucose, even though we do have a glucose example in there as well. Just as a warning, this is one of the questions that comes in quite a lot often, is people are all the time asking us, well, the thing of, what's the accuracy of your sensor in my application? Electrochemical sensors are different from temperature sensors, as an easy example. Electrochemical sensors are in intimate contact with the sample. And this really affects, somebody just brought up the question here about non-specific binding. You don't have a problem with non-specific temperature with a temperature sensor, but with a biosensor, it's sitting in that sample. So if there's a real sample like blood, you could have protein deposition that was trying to confuse your sensor when really you were looking for a very specific type of binding between an antibody and an antigen. And yeah, you have to be pretty smart to be able to deconvolute between specific and non-specific binding. But it's something that, we do. But for every application, I'm always asked, can your sensor work in my application? And the answer is my sensor can, but actually we need a different algorithm every time. So if I make a glucose sensor for a bioreactor, that's one tinyML, let's say, model that needs to go with it. If I make a glucose sensor for blood, that's a different tinyML model. If I make a glucose sensor for interstitial fluid, that's a different tinyML model. So each sensor requires essentially a different, or each application may be using the same general sensor, but it definitely needs a different maths package to go with it each time. And I have made this comment several times, and all I'm saying here is, that this is not university type, biosensing or non-scalable technology. I'm sure many of you are aware of printed electronics. And when I saw printed electronics, when it's been around for, I'm sure, 2 decades at least. But when I saw it, I thought, well, this has been going on for, many decades. And printed electronics to me just remind me of glucose strips. And so we're using, and this is just an R&D sheet, but we're essentially printing often our electrodes. A piece like that, how much does a glucose strip cost to manufacture? If you're at scale, it will cost you something like 2 to 5 cents to manufacture, and it's becoming a very commoditized industry. So I wouldn't suggest you get into it, but it has proven the way we manufacture has proven to be low cost and scalable. It's not the only configuration we work in as well. This is a quick model of the skin. This whole talk is around about the bioedge. So, and it's got, I'm also interested in very much in wearables. So this is a kind of model of the skin with the outer layer, the epidermis, the dermis, and then the subcutaneous layer, which is a reflection of what's going on in the body, for example. So what the 3 CGM manufacturers on the market are doing is they're actually putting essentially the sensing package on the side of on the skin, and there's a filament or wire going through the skin, and at the end of that wire is actually an enzyme, and I'll talk about that enzyme reaction in a minute. So we have these kind of traditional filament or wire sensors. The thing that I'm actually wearing today is and I will talk about this particular device is a microneedle device. So it's not so invasive as the wire, but the microneedles are actually coated with an enzyme and they're undergoing a specific reaction. And we've also worked on measuring sweat as well. Things like lactate in sweat, and surprisingly it worked quite well. I was a little bit surprised because it's a hard application, let's say. And something that we're really quite experienced on is actually making implantable sensors, and I will definitely talk about this sensor package in a minute as well. So the nice thing about the science of electrochemistry, so for me, electrochemistry is the kind of, again, the start of this whole technology stack. Electrochemical sensors, analog front end, digital back end, tinyML has to come into play. And then I'm looking for connectivity, and then I'm looking to deliver my data to the cloud. But the sort of, how do these sensors actually work? Well, the nice thing about this science is we've got lots of techniques. So amperometry, most glucose sensors in the world are amperometric sensors. You apply a voltage, you measure a current, and the current is meant to be proportional to the glucose sensing. Voltammetry used to be a very lab-based technique, but thanks to some of the, I would say, platinum members of tinyML Foundation, it's now chip scaled. We use potentiometry quite a bit. Many of you in your careers have done potentiometric measurements. If you've ever done a pH measurement, at least on a pH probe in a lab, it's a potentiometric measurement. Now, something that's also quite exciting is impedance spectroscopy. Sometimes people call it dielectric spectroscopy, depending on your background, but impedance spectroscopy is a very useful technique. We find it very useful if you're trying to detect proteins through antibody interactions, if you're trying to detect pathogens through binding to surfaces. We really like impedance spectroscopy. And the nice thing is it can all be done now on a potentiostat. This is the finger for scale. And this is not an optimized board, but it's really shrunk down, and it's become certainly much lower powered. So techniques, once upon a time, everyone only ever did amperometry and potentiometry because making the electronics was essentially too complicated. But now actually the problem has now moved from electronics to coding. But we've got enough coders now to be able to make these chips, I want to say dance and sing, or sing and dance for us. So voltammetry, just to sort of give you a sense of, what is going on. This is one of the less used technologies, but I think for the tinyML community, it's a good technique because it's actually very, let's say, feature rich. We can take one of our screen printed electrodes, for example. We can put a sample on there. This is one of the benefits of the science that we do. How much volume of blood does a glucose sensor need? 300 nanoliters. So really, this kind of science doesn't require a lot of sample, which can be very beneficial. And in voltammetry, I mean, I don't want to make this the electrochemical forum, but I'm just saying that all we're really doing is, if you've got an electronics background, we're just changing the voltage and measuring the current in real time. And electrochemistry can be very feature rich. And feature rich is really good for data scientists if they've got many parts of the signal that are proportional to the analyte of concentration. And I'm just running a simulation here. This is starting to look like a fingerprint. And this particular method file is generating, some interesting features that actually develop over time. So this can be extremely data rich. And this is the bit where I said earlier on, once upon a time, we'd have to make a really good sensor in order to kind of give us selectivity and specificity. And now I'm saying, actually, we can use the method to give us lots of features, and we can rely much more on the maths domain than historically we would have been able to do, because the electronics are there, the computational power is there, and there's enough people now starting to understand how to interpret features and to, I don't say, it's not always about AI, it can be neural networks are not good necessarily with the FDA, but we can use data science to help us make better algorithms than historically we would have done. I mean, this is what the raw data from a particular real world sample is, and if we would just do a simple linear regression, but it didn't always have to be simple. It can be more sophisticated, but we really have a signal here, which here is peak height, which is changing essentially with concentration. So I don't want to patronize the tinyML community because you think, well, that's too simple an analysis, but actually that is an analysis that we would do, and it actually, does work. But what makes this actually work really well for us? And one of the things that's kind of held up the whole biosensing thing is actually repeatability of manufacturing. But at ZP, I was lucky. I spent a little bit of my my career in the electronics industry, and that's where I really learned about reproducibility of manufacturing. And because when you look at our data here, each concentration point has actually got 3 sensors underneath it. So we're testing in triplicate, and it's only the reproducibility of our manufacturing that would make this system work, and it's a calibration-free system. So we have to have a repeatable sensor in order to be calibration-free, and we've really focused on making a repeatable electrode system to build upon. I've just talked a lot about voltammetry. Now let me get back to applications. I'm going to talk about embedded applications, wearable applications, and in situ applications, including soil. So case study number 1, this is a veterinary. I will scroll down slightly in case I'm missing any questions. Yeah. So the question was, what kind of algorithm do you use to analyze measurements from voltammetry? I know we are more sophisticated than that, but that was just a simple linear regression. But to be frank with you, we do make much more sophisticated algorithms where we'll actually have a series of algorithms, which one of them says, is our molecule there? Is it in high concentration or low concentration? If it's in high concentration, use this algorithm. And if it's in low concentration, use this algorithm. So that kind of decision tree, I started my career 23 years ago in glucose sensing. That was not going on. Why? Because I don't think we were smart enough. I don't think the computational power was there. I don't think anyone was thinking like that. But actually, you can make a real difference now in biosensing by bringing the way you think to the raw data. So something that's a little bit different at ZP is the people in the lab collecting the data are not the same people who then go on to make our algorithms. I'm sure in your industry, that's also the case, but it's not always been the case in this biosensor world. We're still on catch up. And I will go slightly, I'm not going to say faster, but let me just give you a real world application, enough, a bit of science, but let's talk about this. This is a sensing package from ZP. It does fit into sensing at the Smart BioEdge. What we have here is a package. I wouldn't want it implanted in myself. It's rather big, and it also has a couple of coin batteries in there. So I think the FDA would not allow me to implant that. But it comes in a couple of flavors, glucose and cortisol. And in that package, we've got the transducer, which happens to be piezoelectric because we're actually transmitting the data through seawater. So the electromagnetic spectrum doesn't pass through water quite so well. So we are using a piezo device there. I'm just checking questions, that was all. And whoops, daisy. Yeah, so there's a little bit of a warning here because it's not that gruesome, but just a warning. If you don't like it, if you don't like blood, look away now. Because what we're doing is this fish is not dead. This is a fish, and we're basically inserting the package into this salmon. This salmon is anesthetized. I know I wouldn't have thought that was even possible, but people are willing to anesthetize salmon. And the reason we're doing this is because the second biggest industry in Norway after oil and gas is salmon farming, but 25% of the stock is lost every year. And they want to basically understand the stress on the fish so they can try and reduce the stress on the fish and therefore improve the yield. There's no point building bigger and bigger pens. Let's solve this morbidity problem first of all. So that's the challenge, how to decrease morbidity. You can't improve anything unless you can measure it. Clinically, they think that the glucose and cortisol are the right things to measure. How are we going to continuously measure these things in a fish? Well, Zimmer and Peacock, we love electrochemistry. We think it's very shrinkable and robust. So we put it all into a package, and that package gets put into the salmon, and the salmon is sutured up. No fish were killed in the making of this video. So those fish did survive the process. This one's labeled Soha, who's she's our quality manager, and this one's Evan. He's a bit more, that's my business partner. He's a little slower moving in this particular tank. But at this moment, those packages are actually in these fish. Now, what does the actual data look like? I mean, I don't want the tinyML community, you'll kind of laugh at this. We've got two parts of data here. We've got there's time points coming out of here. So we kind of have these very sort of noisy real data, and through it, then we're sort of, threading the general trend. But that's the kind of role for, tinyML in this kind of thing. I'm sure you're used to this. The raw signal is terrible, but there is information in there. And that for me is the role of, the tinyML community to work with on projects or programs or companies to turn what's not great data into something that's actually useful. And, there was a change in the glucose concentration here, and that was, we were able to validate that by independently measuring the glucose also in fish as well. So it wasn't just a sort of an anomaly, it was a real event. Now the question here is, one of the questions that's being asked here is about physiological parameters such as heart rate variability. What I'm going to say is that at ZP, I'm going to definitely today focus on measuring chemicals in the wet phase. I feel like, ECG, AKG, these things are really done. There's plenty of people are doing this kind of stuff. There is a question here about skin conductance. And that would be the kind of thing that ZP would be interested in, because skin conductance is also skin impedance. So a couple of electrodes on the skin, sure, and you can start measuring the conductivity and impedance and starting to try and make some sort of correlation with, a sort of physiological condition. That would be the kind of thing that we're interested in. But I must admit, in this talk today, I really am talking about measuring chemicals like glucose, lactate, pH, sodium, potassium, gingerol, capsaicin. And that data was gathered over 9 hours. So we were happy to get that. And I'm sorry to jump into bioelectrochemistry for a second, but I just want to do it to sort of, there was a question earlier on about how to stop non-specific binding, or, what about non-specific binding? Now, I am talking about a certain class of sensor here, which happens to be an enzyme sensor. So we do run workshops, but we also do this for a living, where, we'll take a screen printed electrode. We will functionalize the surface with an enzyme and a polymer. It sounds, complicated, but we literally have robots that will do deposition in a very repeatable manner. Then, so that question earlier on about binding, we actually would put like a barrier layer on top. So not everything is necessarily reaching that enzyme layer. In fact, the barrier layer is sort of sometimes designed to be, sort of semi-permeable to the molecule of interest. But for example, a red blood cell wouldn't get through this barrier layer in this case. Now, what's happening is the enzyme, so the molecule here, which is glucose, but I could have also changed this to alcohol. I could have changed it to ketone, but I could have changed it to lactate. There's a specific reaction going on in here. So the glucose, and we're really lucky because nature's given us such a menu of, of enzymes. For every small molecule, there's often, not for every, but there's often an enzyme for it. So glucose reacts with its enzyme, and the enzyme happens to change the glucose. But what's really happening is oxygen is reacting with the enzyme and becoming hydrogen peroxide. And that is what's happening, for example, in a Dexcom sensor and in a Medtronic sensor, if you ever follow the CGM space. Now, that hydrogen peroxide is the electrochemist's friend, and that is a molecule that we can then convert into electrons. So what we're doing is we're applying a potential to an electrode, and the hydrogen peroxide is being oxidized at our surface and essentially giving us electrons. Flow of electrons is current, and the hydrogen peroxide gets recycled to oxygen. So that is the heart of, the CGM, the continuous glucose monitoring space today is approximately 10 billion. I'm not even sure how big the SMBG the blood glucose strip space is, but this is the kind of core science that's going on at the heart of this. And I don't want to trivialize it, but a change of enzyme means you can then sort of change the target molecule. So I'm going to show a change in form factor, but I'm not going to show a change in science. And so when I say a change in form factor, this is a CGM smartwatch. I'm wearing one of the prototypes here. Some of you may not have me up on your screen, but if you maybe watch the recording later on, And you'll see that we have, in fact, I have one of the prototypes and one of the more nicer versions here. I will have some slides on this to now discuss it. So, and I would say that one of the platinum members or sponsors of the tinyML community is instrumental in this technology. We have, it's the same science. So I just want to bring this slide up to just emphasize that the slide I've just shown is actually the same kind of science going on here. And a quick question, a question that just came in is, I am discussing transdermal glucose sensing. I have an entire 45-minute presentation on non-invasive glucose monitoring. So if you ever want to talk to me about Apple's use of photonics for glucose monitoring, I can discuss that at length, but I am talking about transdermal, minimally invasive, if I could call it that, glucose monitoring. So what's happening here is we have this smartwatch. Around the back of it, it actually has an array of microneedles. It's an important design consideration that the electronics and the sensor are two separate components, because people are not going to accept, some of the products on the market at the moment for continuous glucose monitoring, for example, the electronics are thrown away every 10 days. It's just too much of a carbon footprint, and there's also a battery in there. It's not going to be acceptable. But this is designed to have the microneedles engage, let's say, with the watch, but also, which means that we can basically keep on using the watch, but you don't want to reuse these sensors. They've been on the body for some time. It's not a good idea to clean things and put them back onto the body. It's bad business and a risk of infection, let's say. But what's happening in the CGM space is that actually the BGM, the blood glucose monitoring, is shrinking, and the CGM is starting to take over. I think it's generally, there's a real value in continuously knowing your glucose measurement. I think the FDA have started, it's just happened, in fact, the FDA are now approving CGM technology for type 2 diabetes. So far, CGM technology is mostly for type 1 diabetics who really need insulin injections. Type 2 diabetics can kind of control their insulin really through their diet. And now the FDA is saying, actually, you can get sort of reimbursements, etc., for if you're a type 2 diabetic using a CGM technology, and companies in that space are really clearly happy. Do we have a fluoride sensor in our portfolio? Good question, actually. Yeah, fluoride sensor. In fact, we don't have a fluoride sensor in our portfolio. The closest we get to is chloride, but the quick answer to the question is no, we don't have a fluoride sensor in our portfolio. If I Google electrochemistry fluoride, I bet I'll find, the one thing I just want to say, and I'm sorry to make a side comment here, is one of the powers of electrochemistry is there's so much scientific literature, it's quite easy to always find a proof of principle paper out there. So we do really benefit from that. It's the typical thing. There's plenty of academic stuff going on, very little translation. There's that chasm between academia and industry, let's say. And ZP is one of those kind of companies that kind of understands, both the needs of industry, what it takes to make a product, let's say, and also we can read the academic literature and sort of judge whether this is real for real and whether it's translatable into a real product. But the quick answer to your question is no, we haven't got a fluoride sensor. Good question. So just to get back to this particular sensor, so it's, as I say, the sensor itself is something that clicks in and clicks out. It is microneedles. It is electrochemistry. It's not as invasive as the three players on the market today. And if some of you are sort of familiar with companies like BioLink is a well-funded company down in California, in San Diego. They use microneedles. It's that kind of approach to minimally invasive glucose monitoring. If you're not sure what a microneedle is, it's really a small sharp point, something less than 1 millimeter in scale, just enough to go through the dermis, and it sort of, I don't want to say punches holes in, but these are essentially 4 holes that come off microneedles. There's some brown lines going across the screen. Those are the hairs, so you get a sense of the scale of this, and it's not at all painful to do, and it probably, it's more acceptable. It could be more acceptable for some people as rather than the traditional invasive CGM. So there's a whole sort of technology stack of needles going to the skin. There's a chemical reaction, which is the same reaction I was showing earlier on. There's an oxidation of hydrogen peroxides that gives us electrons. We need that analog front end to be able to read the current that's generated. You do need tinyML because a sensor like this, there's lots of noise and artifacts. If I shake my hand, you could appreciate, that can generate noise. So there's a real role for tinyML in this. We do like connectivity, and the data goes to Julie. It's not part of my talk today, but we do have an entire cloud system for receiving this kind of electrochemical data. And I will try to grab hold of questions. So the question here is a good question. I did just touch upon it. There are tens of thousands of articles on electrochemistry, but why are there so few products on the market, let's say? And I think there's just I'm biased. There's a poor translation actually from academia into industry, and Zimmer and Peacock, I think we've got a proven record of actually that translation. But yeah, I see it myself as well. There is a poor translation, but for a company like us, it's a great opportunity as well. So I have talked about continuous monitoring. I am talking about the smart bio edge. So I don't want to restrict this just to human health. So I will play upon global health as well, and just touch upon one of the big applications a big application that's very interesting to electrochemical sensor developers like us is nitrate. There's a big part of the CO2 footprint comes from nitrate production. The Bosch Harbor process that makes nitrates consumes 1% of the global CO2, global energy production. And I want to say something. There is something wrong with nitrate usage in farms, because name me a river in Europe or the USA or near any agricultural center that's not highly polluted with nitrate. I'm thinking about Florida. There's a river, so there's a lake there that's getting, extremely polluted because of nitrate. So there's a problem here because it's using a lot of energy and resources, and they think it's pollution. It causes a kind of global pollution, and locally, you've got something called blue baby syndrome. That happens in the Central Valley in California because there's too much nitrate in the aquifer getting into the water systems. Eutrophication, when all the fish die because there's been an algae bloom and all the oxygen got consumed. Nigeria, acidification of the soil because there's too much nitrate in there. So there's a real, problem with nitrate. Well, I think the tinyML community know this better than most, that actually if you want to improve something, the farmers are not putting the nitrate onto the soil because they think it's a good idea. They do think it's a good idea, but they don't want to waste their money because they're essentially wasting their money. The cost of nitrate is, has really spiraled upwards. But if you want to improve a situation, you have to measure it. Well, there is no continuous measurement of nitrate in the soil. So the farmer is effectively blind. Now at ZP, we have worked on programs where actually we are putting our, and I am watching time here. So we are putting our, let's say, technologies into form factors that allow us to continuously measure the glucose in fish, to continuously measure the glucose in a human being, or continually measure the nitrate in soil. And it's all about connectivity, getting the data, putting the data to the cloud is what we're all about. And I say that, in order to improve a situation, you have to measure it. I think we're the only people with real-time data of nitrate in soil. So the big problem is everyone knows that nitrate's causing a pollution. It costs the farmer money. It gets into the water courses. It's poisoning the water courses. How do we solve it? Well, potentially we will put sensors into the soil. And that's the kind of study that we've actually done. I'll go quickly. I'll talk fairly quickly, which says that we put some sensors into a farm for 3 months. We were measuring the nitrates in real time. The farmer, the large arrow, he adds nitrate to the soil, but he gets no benefit from it because actually the nitrate starts to go down. This is where tinyML can kind of kick in, because actually what's happening is at the same time the soil is drying out. So the farmer has put nitrate on soil, doesn't realize that his soil is drying out, the available nitrate is therefore decreasing because there's literally no water to make it solubilized so the plants can actually get it. At which point it rains. I think tinyML, and AI in general can actually help here. So it rains. What happens is the nitrate shoots up. To turn this data into, let's say, actionable information, actually, it does need AI because the farmer can't be looking at this, interpreting this. But look, it just rained. The nitrate shoots up. So what does the farmer do? He adds a bit more nitrate because he doesn't know. He doesn't know. We know because we're looking at it through our cloud system, but the farmer is blinded to it. So the nitrate's going up all the time, possibly running off into the local river system, possibly percolating beyond the reach of the roots and getting into the aquifer. At which point it is raining all the time. So what does the farmer do? He adds a bit more nitrate, but then he comes into a dry period. So you can almost think about AI doing forward looking and actually looking at the weather and actually saying to him, don't add any more nitrate because actually the weather forecast for the next 3 days, 7 days says it's going to be dry. But he does add some more nitrate, at which point the nitrate starts falling and he doesn't get the return on investment until actually it rains. This data made me realize that this global headline that 67% of nitrate is wasted is 100% correct, because actually he adds 3 doses of nitrate in this particular field when only he ever needed 1, but he was blinded. So look, tinyML community, if you want to improve something, you have to measure it. The measurement starts potentially with biosensors, but probably guys like you that actually turn this into actionable, understandable information. I am going to slightly change course and talk about IVD. I'm also going to try and be a bit quicker because I want to finish. I respect everyone's time. This is just an example of a real world problem, and it talks about decentralizing testing, but it happens to be in the food industry. So this is Professor Richard Compton at the University of Oxford. He understood in his paper that capsaicin is the molecule that causes the hotness of chilies. And the question came up earlier on, and he was using voltammetry to measure the hotness of these chilies. Voltammetry is a great technique. It gives us lots of features. Those features are proportional to the analyte of interest. Give it to a data scientist. They'd love it. They can play with the different peaks and areas under curves and give you a strong algorithm. So what we were doing was we're actually fixing a problem, because in the food industry, to measure the hotness of a product, actually you would use HPLC. It's the same as the clinical system. HPLC is expensive. It's centralized. Essentially, nobody's doing it because food industry is actually a low margin industry. They can't afford $50,000 instruments. They're meant to do panel testing. They don't do it. They're actually mostly doing guessing. And then we have our food sense product, which is really a derivative of the whole thesis about glucose sensing. When you look at our product, we have a meter, we have a strip, we have an application running on a smart device. We had to validate it. So this is really when kind of data science kicks in. We measure it on a golden standard, something that everyone believed in, which is HPLC, and we also measure electrochemically, and we made sure that we were correlating with the HPLC system. We've gone through quite a few iterations, and I'll just quieten that. And now we do have a product on the market. It's been on the market for quite a while. This is one of our engineers talking through it. This video is about 4 minutes long. It takes 3 minutes to actually prepare the sample, and then 1 minute to actually get the result. But that's such a saving on the idea of doing high pressure liquid chromatography measurement. And it's all done using the same electronics that I've been talking about throughout. So the same electronics to do the glucose sensing, same electronics now to do the chili sensing. We're always using the same electronics. We're just recycling, let's say. And then in real time on the phone, the person gets the Scoville heat unit measurement of that particular chili. Chilies is one of the ones that takes the most sample preparation. This is a true platform from us. We realize now that, we actually got a platform. So people come to us. I say, look, let's get you to a minimally viable product pretty quickly. So we take a sensor. We have electronics. These electronics can essentially do any of our methods: amperometry, impedance spectroscopy, voltammetry, potentiometry. We can quickly skin the apps. The apps make the meter do the work. The sample goes on the sensor, and the app makes the meter do the work. The meter gives the data back to Ivory as that Danilo is kicking in, so I will be quick. But eventually, we actually get the concentration. So And I'll just finish with a final one here, which says it's not all about food quality. And here we're actually doing a real-time measurement of nitrate, which I think is significant in water quality. So I think my call to action is this. It's a bit presumptuous of me, but all the easy stuff has been done. And actually the hard stuff is, it's time to work on biology, but let me extend that and say it's time to work on biochemistry and chemistry. So I just want to say, thank you. And thank you so much for allowing me to talk rather a lot today. Thank you so much.
Excellent, Martin. Thanks so much. But me personally, I learned a lot from your talk. And as promised, I would have a question for you. I know that the time is really tight, and so people, especially in California, is going to leave. But here the question. I saw glucose sensors on the market, and I'm not an expert. I'm not working on it, but it touched me the temperature range. It was very tiny, few Celsius degree up to 28 Celsius degree. And the sensor cost remarkably. The cost is very high and is disposable, Then, I'm spending vacation in Sardinia. Sardinia is a large island in Italy. I am in the south. And by chance, the temperature was 42 degrees in my car. And one colleague of mine said that he experienced 47 Celsius degree. And I cannot imagine how, I didn't measure how high was the temperature on the beach under the sun at noon. So the question is, how can, for example, for a glucose sensor, but any other you think is more urgent the issue, how repeatable, how reliable, is the measure? It's just a matter of better material, better manufacturing processes, and is there any opportunity for the tinyML community to develop solutions that complement better material or better manufacturing process?
I think the tinyML community, you've got two questions there. One of them is about manufacturing, and one's about temperature. So I'll go to the manufacturing first of all. I said it in my talk that if you use, so the tinyML community can definitely help on the manufacturing side, because I think that if you use more sophisticated method files to generate the data, you will get features that characterize the sensor in situ. And you can use those features to then normalize the signal. So at the moment, you have to make a brilliant sensor so that the whole batch is completely the same. And therefore, when you go into the field, it's essentially calibration free. But I actually think that you can use more sophisticated excitation methods on that sensor to say, is my sensor a class 1, a class 2, a class 3, and then treat it with a different algorithm depending on that classification. So the tinyML community can help on the need for manufacturing, repeatability of manufacturing. That's one thing. Your other question about temperature. If the sensor is in the body, then the body's pretty good at controlling temperature, but enzymes are temperature sensitive, first of all. And I also think that the electronics are actually going to be temperature sensitive. So you probably do need to know about temperature in those electronics, because if you're certain that all your components are sort of temperature insensitive, great. But if you're not, you better measure it and compensate for it.
Okay, thank you very much. Really, we'll look forward to discuss more with you, because there are other things worth to discuss, like availability of data, and to let the community to share advancement in such respect, models. But maybe we are going to run out of time. So a few things, Martin. There are, if you see other questions that are unanswered, please address them through the tinyML forum. So because it was very interactive, it was a great talk, a great also way to talk with the community and the community with you. So definitely your answers will be more appreciated also on the forum, as well as they were during your talk. we're going to run out of time due to recording issues, so I'm going to request the remote control to you so that I will close your talk. But thanks so much. It was outstanding to have your time, your experience on biosensor, which is great, and it was very informative for the community and for me as well. So thanks again thanks you so much for your time, and let me go on the remaining slide. Okay, so any tinyML talk like the one with Martin can happen thanks to the great support of our strategic partners, which I listed before, and they are very committed to help the community with this type of event, with many others, and also providing support to make sure that the content as generated today with Martin is available for the community to benefit. Okay, so let me go through the executive strategic partners to remind who they are. Qualcomm, Sentient, the Platinum strategic partners, Aetrios, Sony with Aetrius technology, the Gold strategic partners, Analog Devices, ARM, Edge Impulse, Infineon, Innatera, Microsoft, Renesas, STMicroelectronics, Synaptics, the Silver strategic partners, quickly reminding, AEI Zip, Arduino, BrainChip, Efficient, Greenway Technology, Groverty, IMAX, IBM, ImageBob, Not AI, NXP, Polytechnology, Quixo, Schneider Electric, Sensimil, Silicon Labs, and TDK. As usual, the copyright noise. Thanks so much, Martin, and talk to you soon. Bye to everyone.